Welcome back to Mad Medicine. In this lecture, we're going to be discussing the concept of cellular damage. Now, I'm sure you have a very good basic understanding of cellular damage. It's not that difficult, but for your exam purposes, you have to have a very comprehensive understanding of how cellular damage occurs and what really encompasses cellular damage. So with that being said, we're going to be discussing this topic in detail today for your exam prep purposes. Let's first start off by talking about cellular injury. Now, the main thing you need to remember is our cells are able to handle a certain amount of stress, but when the level of stress exceeds our cell's ability to handle that stress, you are going to get cellular injury. So, cell injury occurs when cell stress well exceeds cell's ability to adapt. And there are many different ranges of injury, and they all depend on the type of cell, the type of stress, and the severity, more importantly, the severity of the stress itself. You have certain cells in your body, like neurons and cardiac cells, that are very susceptible to stress, meaning they are more likely to get damaged and die when they're stressed out, whereas other cells, like your muscle cells, are not. Your muscle cells are able to handle quite a lot of stress because they're able to maintain function in a low oxygen state. Now, there are many causes of cellular injury, like hypoxia, inflammation, malnourishment, even genetic mutations, as well as trauma. Most of these we've discussed in our previous lecture, so make sure you check them out. But let's get a quick recap of hypoxia, because this is by far a very important concept of cell injury, very important mechanism, of cell injury. So, what is hypoxia? Essentially, it is a condition that occurs when you do not have enough oxygen supplying our tissues. This is a decrease in oxygen at the tissue level. Okay. Now, when you have low oxygen being delivered to your tissue, you are going to have hypoxia. That's essentially its TLDR definition. Okay. Now, the tissues are dependent on oxygen because oxygen is the final electron acceptor in the electron transport chain, meaning it helps promote ATP production. You need oxygen to produce ATP. Okay, and a lack of oxygen therefore will lead to a low ATP production occurring, aka decrease in energy. This is a very basic concept, very basic review of hypoxia. We have a whole lecture dedicated to hypoxia. In fact, we have 2 lectures dedicated to hypoxia in 2 separate parts, and I highly recommend you watch them because this is a very high yield topic, something that you will be tested on no matter what at every stage of your medical education. So that is the concept of hypoxia in a nutshell. Very straightforward, to the point. But now we got to talk about cellular damage, because both of these concepts are going to lead to cell damage. If you have a state of prolonged hypoxia, our cells are not going to be able to maintain adequate processes, adequate mechanisms, because there's not enough oxygen, and eventually you will run out of ATP, and that is where we are going to see cell damage occurring. Okay, ATP loss or ATP depletion causes our cells to get damaged. So let's talk about cell damage really quickly. A lack of ATP can occur in 3 main mechanisms, okay? It can lead to 3 main mechanisms of cell damage. The first is blocking the sodium-potassium pumps. When you block the sodium-potassium pumps, you do not have the ability to have your sodium leave the cell. And what happens when there is too much sodium inside of the cell? The osmolarity will increase. And when you have increased osmolarity, the solute is going to be high. That means the solvent has to increase. Therefore, you are going to get cellular swelling occurring, and eventually the cell will burst, okay? Because of the fact that you have so much sodium in there, the cell has to essentially burst, okay? That's what ends up happening if you block the sodium-potassium pumps. Now, there are other consequences if you block this. Your potassium level could get increased extracellularly, which can affect the heart. It can cause a lot of other problems in your body. That's one thing that low ATP does. Another thing that low ATP does is that it blocks the calcium pumps. Now, you see, our cells want to have low calcium in the cytoplasm, okay? And when you have low ATP, you are going to have high calcium in the cytoplasm. Now, why do you want low calcium? Because calcium activates enzymes, and those enzymes are very detrimental if they are overactivated. Because once they get overactivated, they can cause cellular damage to occur. These enzymes can essentially degrade the cell itself, and that's why you want to have low intracellular cytoplasm calcium concentrations. Okay. Now, that's the second way. So first way, you are going to block the sodium potassium pumps. The second way is blocking your calcium pumps, okay? And then the third way is by activating anaerobic glycolysis. This is going to lead to a buildup of lactic acid. And lactic acid, as its name suggests, has a very low pH because it is, you guessed it, acidic. Lactic acid is acidic. And when you have something that's very acidic, it's also going to lead to cellular damage no matter what. Okay, so this is the basic concept of how, the basic mechanisms of how cell damage occurs when you deplete ATP. When you deplete ATP, you're going to block your sodium potassium pumps, you're going to block your calcium pumps, and you're going to cause anaerobic glycolysis to occur. If you block your sodium potassium pumps, you're going to essentially cause increase in sodium intracellularly, which is going to cause the cell to swell and it will die. If you block the calcium pumps, you are going to have high calcium concentration in the cytoplasm. And when you have high calcium concentration, you are going to activate intracellular enzymes more than they need to be, and they are also going to cause cell damage. And then finally, with anaerobic glycolysis, you are going to build up lactic acid, and the acidic environment will damage the cell, leading to cell damage. So that is what happens. Now, there are two main characteristics of cell damage that you need to be aware of, okay? And these characteristics are very important because they are essentially they essentially describe the process of which cell damage is occurring, how far progressed the cell has been damaged, okay? So let's talk about the first process, or the first characteristic. That is cellular swelling. When the cell swelled, like let's say this is a normal cell, okay, this is the normal nucleus and whatever organelles, and the cell swelled larger than it normally should be, okay, this is initially the first phase of cell damage. Okay, the reason why this happens is because you have low ATP. And when you have low ATP, like we talked earlier, you are going to prevent the proper function of the sodium potassium pumps. And that is why you are going to have high intracellular sodium concentration, okay, which will cause increase in H2O going into the cell. Okay, this is an arrow pointing into the cell. Horrible arrow, my bad. But that is the reason why you get cellular swelling. The key thing to remember is that this stage is reversible. Very, very important. You can go back from the swollen side to the normal side by essentially producing ATP and activating your sodium potassium pumps to get the sodium outside of the cell. What else happens during this time? Essentially, you are going to see loss of the microvilli because you are expanding the cell. It's swelling, so microvilli will go away. They get essentially eroded as the cell swells. The membrane will start blebbing. The rough endoplasmic reticulum will swell, and when that happens, because the rough endoplasmic reticulum is swelling, you are going to get a release of the ribosomes from the rough endoplasmic reticulum. When also that happens, you are going to get a decrease in protein synthesis. All of this is connected. Very, very important, okay? This is very important. Now, this is the initial stage. If you do not reverse it at this stage, we are going to progress to the membrane damage stage. This usually happens because of long-standing cell damage, okay? This is not a short-acting phase. It happens after a long time when the cell is being pushed way past its limit. When that happens, you are going to see the membrane damage happening not only to the cell, but also to the mitochondria and the lysosomal membranes. And this is very important, okay, because the mitochondrial and the lysosomal membranes carry intrinsic factors that will destroy the cell if those membranes are damaged. So that means the membrane damage is occurring across the cell inside the cell, not just in the cell itself, but in the organelles of the cell. This is going to be irreversible. And the characteristics of this is by far things you're going to see when cell damage is occurring across the board. So you're going to see an increase in the intracellular enzymes, such as troponin, when you have cardiac leak or even non-cardiac troponin release, right? You can see ALT, AST from your liver cells. If your liver gets damaged, you're going to release those liver enzymes, and that's how you know that the cells are being damaged, okay? You're also going to see an increase in the intracellular calcium in these cells specifically. Now, this is not really useful clinically, but something you need to know for the test. You're also going to see cytochrome C being released because cytochrome C actually sits in the mitochondria. And when the mitochondria is damaged, you are going to see cytochrome C in the cytoplasm. And when cytochrome C goes into the cytoplasm, it activates a cell cascade which leads to apoptosis. This is a pro-apoptotic factor when it is found within the cell mitochondrially. And then finally, you have the lysosomal factors in the cytoplasm. If you recall, the lysosome carries all these enzymes, and when you damage the lysosome, you are going to damage the ability to essentially retain those enzymes in a closed-off environment. You are going to free these enzymes, and they're going to go and damage the cell however they so please. Combined with the high intracellular calcium, these factors will be even more activated, and they are going to damage the cell very, very well. So let's talk about this, a quick recap really quickly. So when you are damaging your cell, essentially you are having this process take place because of low ATP. When you have low ATP, you're going to block the sodium-potassium pumps. When that happens, you initially go to a cellular swelling stage. And this stage right here is a reversible stage, okay? Now, when you block the sodium-potassium pumps, you have high sodium intracellularly, and that causes the cell to swell because water follows sodium. Once that happens and you don't remove the stressor and you don't activate ATP production, you are going to progress to the membrane damage phase where you are going to see long-standing damage of not only the cell membrane, but the mitochondrial and lysosomal membranes as well. You're going to release factors like cytochrome C and the lysosomal factors, which are going to destroy the cell. You are going to see activation of other enzymes because you are also going to have a high intracellular calcium concentration because the calcium pumps don't function either. Very straightforward, but it can get complicated really quickly. I highly recommend you watch this video again if it did not make complete sense the first time, because there is surprisingly a lot of information, even though it can be very intuitive and it's very simple. It's just a lot. Okay, so with that being said, I hope this was helpful for you. If you like this content, don't forget to subscribe to our channel because your support means a lot to us. And if you want to see more content like this, go to our website, www.madmedicine.org, where you can find additional free educational content for your studying purposes. Enjoy.